As we move toward the end of the semester, we finally get to talk about speciation and extinction. And these two processes are actually also integrally important to evolution. For example, the idea of speciation, where new species come from, how do species arise or branch off from pre-existing species? And on the flip side of that, what happens when a species ends? What causes that? What contributes to that? Are there factors that we should take into consideration about what makes a species more likely to survive a mass extinction, or what makes a species less likely to survive? So that's where we're going in this particular joint chapter. I've merged them together to try to keep this material a little bit shorter. Not all of the topics are actually going to be discussed that are available in your textbook. You're only responsible for the parts that I do talk about. So one of the common things that I hear against evolution is, but nobody has ever seen a totally new species evolve. Actually, yeah, we have several times, including fairly recently, and including several times in written human history. So this particular example, I'm going to kind of leave it up to you to make a judgment call about this. But what we're looking at is an image of the London Underground subway tunnels, and they started construction on those during World War II. As they built these things, these populations of mosquitoes actually kind of migrated into these tunnels where they adapted to the warmer and moister environment underground. And these became known as Culex pipiens, regular mosquitoes, but these were molestus versions, as in they molest us, they bother us, they annoy us, they are pests. So here's what you should actually understand about these particular mosquitoes. Compared to the regular Culex pipiens, the regular mosquitoes breed above ground and the molestus breed below ground. Okay, well that's a matter of geography. I'm not totally convinced that that's actually how we should define them as a separate species, so not totally convinced at this stage. The regular mosquitoes tend to mate in open spaces, whereas the molestus mate in confined spaces. Again, how convincing is this? This is just a factor of being underground. It's just a slightly different environment. Here's where things start to get a little bit more interesting, though. The regular mosquitoes preferentially feed on birds, whereas the molestus mosquitoes feed primarily on mammals. Now, this actually goes a little bit beyond just the fact that you don't see that many birds living underground. This actually is, even when they test this like in a laboratory setting, the Culex pipiens molestus actually prefer to bite mammals. That's annoying. And that's one of the first really, I guess, biological differences that I can see out of this data set. Their egg production is actually also different. So regular mosquitoes, the female mosquito bites the host animals, whether that's a bird or a mammal, in order to get a blood meal. And it uses that energy, that glucose and those proteins, to actually fuel egg production. So the female mosquitoes specifically bite so that they have enough energy to lay eggs and contribute to the next generation of mosquitoes. In the regular pipiens, they actually require that blood meal, but the underground molestus versions don't anymore. They just bite us, I guess, because they can. I don't know if I've mentioned this, but I think mosquitoes are horrible little things, and I kind of wish they'd disappear from the face of the earth. No such luck, and apparently now we have them in subway tunnels. Last but not least, the regular Culex pipiens go dormant in the winter when the temperatures fall and it's cold outside, and the molestus do not. They are active year-round, so you're just as likely to be bitten by a mosquito in February as you are in July. That's just terrific. So, my question to you guys in this next slide is, what do you think? Do you actually think that these mosquitoes should be considered a fully separate species at this point? Yes? No? Not sure? Let me know in the next slide. So one of the things I didn't tell you before you answered that question is that the Culex pipiens molestus mosquitoes can actually no longer interbreed with the regular Culex pipiens mosquitoes above ground. Even if you put them together, they don't mate with each other, they don't produce viable offspring. That's going to become important in a little bit as we start to look at maybe how we define a species. So in this chapter, what we're going to be looking at is what is a species? I mean, how do we define this idea of a species? How does this speciation event occur? When does a species end? What contributes to that? All of those types of questions. Well, maybe not all, but some. What creates reproductive isolation among populations, and how is this intertwined with the idea of speciation? When we get to the extinction part of this, what is background extinction and how is that different from mass extinctions? Are we really looking at a mass extinction caused by human activity right now is one of the topics in this chapter as well. Is the rate of evolutionary change sort of slow and steady, which is kind of how we've been thinking about it, or does it actually have periods of rapid change? And again, are there some particular characteristics that make a species more likely to go extinct? So that's where we're headed with this information. So our first question in this is, what is a species? How do we define a species? And this is a problematic issue to some extent. How do you define when a population becomes two distinct subspecies instead of one original species? To rephrase this, wolves and dogs. When does Canis lupus familiaris become a separate species, dog? And when is that fully separate from wolf, Canis lupus? So the evolutionary definition, since this is an evolution class, states that a species is a lineage of populations which maintain its identity distinct from other such lineages, and they have a shared past and will have a shared future on a phylogenetic tree. Great. I don't know about you. I don't carry around phylogenetic trees to check and see if something should be considered a separate species. In evolution, if you're looking back at things and if you're looking at phylogenetics, this can actually be really useful, but it's not practical on a day-to-day basis, and it certainly doesn't really help us come up with something that's kind of quick and easy. So we need something that's easier to apply, something that's inherently simpler. And that brings us to our other two main concepts for species definitions. One of these is the phonetic species concept, and this is really technically a statistical method. It's based on the number of similarities between individuals. So this is kind of how we naturally tend to group things, in my opinion. So we know that there are lions, we know that there are tigers, but we don't see that many crossbreeds. We don't see that many ligers or tigons. Yes, there's a difference. We'll come back to that. Because it's technically considered a statistical method, there's a reason behind that. And what that really amounts to is how many similarities are enough? How many differences do you actually need before you say, okay, these two things are really separate things? And that's where the math helps us to draw clearer dividing lines than just us trying to eyeball things. However, inherently, I think that most of us tend to use the phonetic concept kind of just out of our natural understanding and observations of the world around us. The other definition is considerably more broadly accepted, and this is the biological species concept. So this actually says that a species is a group of actually or potentially interbreeding individuals that are reproductively isolated from other groups. In addition to that, two organisms have to be able to interbreed and produce viable and fertile offspring. So their offspring have to survive and be capable of producing more offspring themselves. So if we go back to the question about the dogs and the wolves actually continue to mate today, and they actually produce puppies together. Yay! And there are actually people who keep these as pets. You have to get special licenses for that. You can't just do it on your own, or legally you're not supposed to. So they produce viable offspring. Those offspring are fertile. They grow up. They can interbreed with dogs, wolves, other crossbreeds. There's basically no dividing line there. So this is why we actually consider technically dogs and wolves to still be the same species. That's different than what happens with ligers and tigons. The ligers and tigons, although they're a crossbreed of a lion and a tiger, they can produce offspring. It's not super common, and those offspring can't produce offspring in turn, so they're usually infertile or sterile. So this is workable for existing species, but it tends to be somewhat impossible for fossils. I used to actually say that nobody had ever seen two fossils going at it. but that turns out to be wrong. So here are two kind of interesting examples. The first one on the left is actually a couple of turtles that were caught during the mating process. They died and they were actually fossilized, still in mating positions. So because I don't expect you guys to be experts in fossil mating of turtles, here's what you're looking at. This larger turtle on the left-hand side is the female turtle. The smaller one is the male turtle. That's actually consistent with what we see in modern turtle species today. And their positions, the way they're actually sitting, corresponds to modern turtle mating positions. In fact, they can actually see kind of the tail position of the male turtle. It's kind of coiled and wrapped under the female, just like in modern turtle mating. So that one's kind of cool. And in the second image, we actually have insects that were caught in the process of copulation, and they died that way. And this is a really rare event. There aren't that many good examples of fossilized mating. So obviously, we would guess, based on this information, that the turtles are of the same species and that those insects are of the same species because they're mating with each other. But I refer you back to dogs. How many times have you observed or seen YouTube videos of dogs attempting to mate with wildly inappropriate objects? Yeah. Okay. That's a normal behavior. In fact, it occurs in fish, it occurs in almost all species. It certainly is nothing in particular against dogs, but assuming that these matings were able to produce viable, fertile offspring, we would technically consider these to be the same species under the biological species concept. And so this one is really kind of the easiest to apply. We can observe mating, we can see whether things can interbreed with each other and produce offspring. That's easy for us to track, especially for living organisms. But in the case of like fossils where we don't have fossilized mating going on, the phonetic species concept is still largely used today. And again, it's based on the idea of the number of similarities as how we should define a species. So for example, we have lions and tigers and cheetahs, and these form very distinct and easy to identify populations to us, even though they're all big cat species. So they tend to kind of cluster in phenotype space. Again, this clustering, it's something that we actually have to run math on and try to solve statistically before we're really comfortable with that, but historically, we didn't bother. So some of both can actually apply, but the biological species concept actually requires that our populations become eventually unable to breed with each other for whatever reason, and we'll talk about how and why that can occur. But just to start off here, what we've got are a bunch of islands with a bunch of different critters on them, and let's say that population 1 over here can actually functionally interbreed with population 2. These arrows are supposed to represent gene flow. 2 can interbreed with 3, 3 can interbreed with 1, 3 and 5, 2 and 5, so all of these can interbreed with each other and produce offspring. Voila, we call that a species. 4 and 6 can interbreed with each other. That's why we consider those to be a species. 7 is off all by itself and can't interbreed with any of the others, so in this particular example we would have 3 fully separate species. Okay, this can be due to geography, like oceans and islands and just sheer distance. It can be due to mountain ranges and rivers or even highways, for example, or it can be due to genetic causes or other factors that influence whether or not 2 species can actually mate and produce offspring. So on the larger scale, what we're looking at is how that reproductive isolation can play out, and in particular in relation to things like geography. So in our first example, we have allopatric speciation, and this is where your 2 populations become separated due to geography. They are completely physically isolated. They can't really physically encounter each other, so they don't mate with each other. They could be on often like different sides of a continent, East Coast versus West Coast. They could be on different islands. They could be on different sides of a river, whatever it may be that prevents them from physically interacting with each other to mate. And there are 2 ways this in particular can play out. The first idea is the dumbbell model, also called the vicariance model. And in this particular model of reproductive isolation and allopatric speciation, what happens is a geographic barrier separates your population roughly in half, and that's the biggest difference here. This one is basically an equal division of your population, And over time, the two populations gradually become genetically different due to different selective forces, genetic drift, non-random mating, all of those things that we talked about earlier this semester. This is what's going to be driving those two populations to become different over time. In the second method of allopatric speciation, you have the peripheral isolate model, where a small subgroup is what gets separated from the main group of individuals. And that small subpopulation ends up in an isolated region where it genetically becomes distinct. And in this particular case, because it's a smaller subgroup, what we would reasonably expect to see is a lot more genetic drift, a lot more inbreeding, so we would expect to see reductions in heterozygosity, fixation of alleles, and eventually these populations should become genetically different enough that they can't interbreed anymore, even if they physically mixed. So that's allopatric speciation. Allo meaning different, different lands. They are in different environments. Our next model of speciation isn't quite as clear-cut, and parapatric speciation are basically in kind of adjoining geographic regions. The animals or plants or whatever you're interested in, insects for example, may encounter each other, and they may even successfully mate, but usually only when within a very limited window of geography, a very limited territory where they actually exist. And we typically call that area right in there the hybrid zone. And that's a big tip-off for allopatric speciation. You have a species over here and a species over here, and where the two species meet, there's limited amounts of interbreeding going on, but they're fairly distinct. Furthermore, those hybrid individuals tend to only really be able to survive well in that one kind of geography range area between those other two groups. So it's usually due to some really weird selection going on in that particular area that favors the heterozygotes, that favors the hybrids of those two parental groups, where they can't really survive outside of that area. But hybrid zone is your big tip-off for parapatric speciation. Sympatric speciation is the toughest of the three, and that's because there is no geographic reason. In fact, these critters, whether they're like birds living in the same forest or fish living in the same pond, they actually encounter each other. And they could potentially mate with each other, they just don't. So this is thought to be due to competition for resources. And as it turns out, Darwin's finches are a really good example of sympatric speciation, although that's not the example I'll be using. What happened with Darwin's finches, if you are familiar with the story, they had surprisingly marked differences in their beak sizes and shapes, and this was because they were eating different types of seeds and nuts. So those differences in food sources gradually drove those bird populations apart, and they quit interbreeding with each other. Eventually they became fully separate, distinct species, even though there was no geographic barrier, and they were just sort of all mixed in there in the Galapagos Islands together. So that's sympatric speciation, same territory. So our first example is actually in shrimp, and you don't have to be experts in shrimp mating or anything like that. But what we're looking at is starting about 10 million years ago. The North and South American continents actually weren't separated. The water levels were high enough that the shrimp in this region could actually freely mix and interbreed with each other. So, according to the biological definition, they were the same species. As the planet cooled off a little bit over the next 5 million years, sea levels actually dropped, and this exposed the land bridge between North America and South America, the Isthmus of Panama. And that started forming really about 9 million years ago. So we've had almost 10 million years of separation between the shrimp on the Pacific side of this land bridge and the Caribbean side of the land bridge. Shrimp are fairly small little critters. They are not going to swim all the way down around the tip of the South American continent, back up the other side, and over to the Caribbean side just to find a mate. Never gonna happen. So they became isolated. And this was a pretty equal split. I mean, you've got plenty of territory, plenty of shrimp on either side. So, do we actually see differences? Have they become different? Well, they took a bunch of shrimp from the Pacific side that were of different species, and they found out that 60% of those shrimp could still mate and produce viable clutches. So I guess there weren't so many separate species. And they did the same kind of study over on the Caribbean side. They got shrimp from different species, and they could still interbreed. So functionally, we were maybe incorrectly calling them separate species of shrimp. But what's interesting is what happened when you tried to put Pacific shrimp and Caribbean shrimp together. So they actually did some genetic analysis based on mitochondrial DNA from the shrimp. Seems like a lot of work. But they found that these actually did form natural sister species. So, for example, C6 and P6 up here, those actually formed a sister clade. They were genetically similar to each other, and C6 was more similar to P6 than it was to any of the others, like even C4. So that suggests that they became separate species because of that land bridge. And in fact, we've got the 6s, the 3s, the 5s, the 4s, the 1s, the 2s, and the 7 different sister groups here. And they actually observed behavioral differences. So the Pacific shrimp and the Caribbean shrimp, one group was actually more aggressive than the other, and there were differences in mating behaviors as well. I don't remember which ones were more aggressive. It doesn't particularly matter. I think the Caribbean shrimp should be the ones that are more cool and more chill because, you know, all the rum, right? Bad joke, I know. So when they put these sister species, according to phylogenetics, together, they actually found out that only 1% of the matings were viable. This is a very low percentage. So basically this supported the land bridge hypothesis and gave us a pretty good idea that in fact, at least for these 6 groups, that these were functionally basically separate species. So in this particular case, you had 1% that were successful matings, and since the P7s down here, that actually would have been one of the ones that was like 60% successful matings. So that was allopatric speciation and a real-world example. The peripheral isolate model, we get to go back and revisit the spruce trees that we've talked about before. So the red spruce and the black spruce are related species. You find the black spruce in Canada, the red in the United States. Black is thought to be the ancestral, the older version. As we talked about before briefly, the seeds are kind of distance limited. They're bigger, they're heavier, so they can travel a little ways, but then they just kind of fall. Pollen, on the other hand, can circulate a lot more globally. So what happens is that when you look at the populations of these trees, what we find is a whole lot of black spruce up in Canada, and we find red spruce tracking along the Appalachian Mountains in the US. And that's why you see those differences. Now, when you look at the genetics of those populations, especially the mitochondrial DNA, because seeds don't spread by wind nearly as easily, what we find is that almost all of these subpopulations in these regions, the red spruce, have basically mitochondrial subtype 4, 4, 4, according to the key on this, 4 with a little bit of 3, so these are both 3 and 4 to some extent, whereas the black spruce had a lot more genetic diversity. So in this particular first one, we had some type 1, 2, 3, and 4, same over here, and even in population 3. So how this played out. Initially, when the planet was considerably colder during the last ice age, yes, think of Scrat for a moment. During that last ice age, a huge amount of the US was actually covered by iceberg. And as that retreated, as the planet got warmer again, as the glaciers retreated back toward Canada, they actually dropped seeds from these spruce trees. And those seeds sprouted and grew all through the Appalachian Mountain region. And as it turns out, most of them were from mitochondrial subtype 4, maybe with a little bit of 3, so probably from that population, most likely. They were reproductively isolated. They couldn't, trees can't pick up and move to mate with other trees. The seeds are limited by distance, so they were reproductively isolated, and it also confirms the idea that these populations had increased genetic drift and fixation, because if you look at these lower sets of samples, they were all fixed for mitochondrial subtype 4. Cool. It all ties together. I love it when that happens. So parapatric speciation occurs where you would have a hybrid zone, and they actually looked at this in particularly the sweet vernal grass. This is a summer grass. It's kind of a weedy looking thing. You've got a picture of it there. And they compared the versions that grow in pastures to the versions that grow in mining areas. And in particular, they were looking at the ability of these plants to absorb zinc from the environment. Some plants are actually surprisingly good at sucking heavy metals out of the environment. In fact, sunflowers can absorb up to 70% of their body weight as lead. So they're actually used to clean up some heavy metal spills. No, we don't get to eat those sunflower seeds because toxic. So how do these plants do that? They've got some biological quirks that enable them to survive these high zinc or lead levels and maybe even thrive in those areas. In the pasture regions, what we actually see is very low zinc tolerance. In the mining regions, we see significantly higher zinc tolerance. And basically in this hybrid region, we see something in between, an intermediate tolerance to zinc. The hybrids of the pasture and the mining grasses actually do well in that particular region where the zinc levels are also sort of intermediate. Very good example. Another example that gives us parapatric speciation and a hybrid zone that's pretty clearly defined. They looked at mountain sagebrush, and in particular, they looked at this growing in 3 different regions, and they looked at a couple different species. So they had Artemisia tridentata, and Artemisia tridentata vassiana. So I'm just going to call them tridentata and vassiana to help keep them distinct. So in the basin regions, the fitness of the tridentata versions was high, and the fitness of the vassiana versions was pretty low. In the mountain regions, this was exactly reversed. The vassiana had good fitness, the tridentata had crappy fitness. But what becomes really interesting is what happens in particular when you look at the hybrid versions of these particular plants. In the basin region, the hybrids didn't do so well. They did okay, but not great in the mountain regions. But in the in-between territory, the hybrids between the vassiana and tridentata did the best, right in the middle. So to put this in perspective for local people, here in Virginia, we have everything from seashore, right here in Hampton Roads, up to the mountains in the Appalachian Mountains. We've actually got a pretty good range of elevation that goes all the way through the state. This would be like trying to get trees from down here and trees from up in the mountains to mate, and their best scenario might be somewhere around the middle of the state in terms of where they could survive the best, if they were able to produce functional offspring at all. Ring species are another case that is sort of a blend. It's a weird model, but it actually occurs in nature. And in particular, it occurs around ecologically unfavorable habitats where there's problems with survival and reproductive success because the environment is kind of poor. So in this particular case, you're looking at a bunch of salamanders, and this is around a particular lake. And so you've got species 1 or group 1, population 1 up there that can actually interbreed with population 2, 2 can interbreed with 3, 3 can interbreed with 4. So at least at this point, we would call all of that a species. Population 2 can interbreed with 5, 4 can interbreed with 9, 5 can interbreed with 6, yikes, 9 can interbreed with 10, and so it goes all the way down the lake until you get to the bottom edge of the lake where the populations 8 and 12, although they actually encounter each other, they don't interbreed anymore. They have become different enough that they no longer mate. And there's a lot of questions for how and why could this happen. I mean, this is just around a lake. It's not like they're separated by an ocean or a continent or something. Well, It could be biology. I mean, there could be genetic reasons that they're incompatible. It could be something as simple as maybe they developed different mating rituals and they don't find each other sexy anymore. Remember, mate attraction is a big part of this too. So whatever might be the reason, we've actually observed this for not only salamanders, but also songbirds around the Tibetan Plateau. These ring species actually do occur. And you end up, at least temporarily, with small areas where you do end up with hybrid zones. So that's kind of why it's listed in between this whole with the parapatric information, sort of. But we're about to go to sympatric, so there's a little bit of that too. But it illustrates everything that we've been talking about. The biological species concept of interbreeding, brief periods where or places where these salamanders can mate and interbreed with each other, and eventual reproductive isolation. So our last example of these speciation mechanisms is sympatric speciation, and this one is from Lake Apoyo in Nicaragua. And Lake Nicaragua is a very large lake that's actually basically kind of over here at the edge of Nicaragua, right where it kind of borders on Costa Rica for part of it. And it's actually only separated from the ocean by a small little piece of land that's about 50 miles or so wide. So that's not a lot of difference, but it's enough that fish aren't really going to go crawling out of the lake and over into the ocean. This is a freshwater lake. And if you actually look at the shape of the lake and you look at the picture of the lake, you can kind of figure out that this lake looks like maybe an impact crater. I mean, those walls are kind of steep. So it is a geographically isolated kind of situation. That's not really the point. The point's going to be what goes on within this lake. And within the lake, we find two kinds of cichlid fish in particular. We have the Amphilophus citronellus, which are these kind of bright orange fish. They're called the Midas cichlid because of their golden color. And these fish in particular tend to eat biofilm. Biofilm forms a fairly hefty chunk of their diet. They also eat a bunch of algae. That forms another big chunk of their diet. And the rest is kind of made up of other fish and some insects. On the other hand, the other fish are the Amphilophus zaliosus fish, as in zebra striped, which is exactly how they got their name. And these fish primarily eat biofilm. There's a smaller part of their diet that's insect-based. They also have a little bit of fish and maybe a little bit of zooplankton, and that's part of what separates them. They also live at different levels in the lake. For example, the citronellus is actually labeled as a bottom feeder, and the zaliosus is actually labeled as an open water forager. So they actually don't exist in the same depth and the same level of the lake, and they don't actually intermix with each other that much. What's kind of interesting about this, though, is that the algae primarily has to grow near the surface of the lake where the sun penetrates, so I can't help but wonder if the labeling on these is actually backwards. So one of these groups is better adapted for surface conditions, the other is better adapted for conditions in the depths where, frankly, the zaliosus would actually be better hidden and better camouflaged. They eat very different diets, and they don't interbreed with each other. They occasionally encounter each other, they're just not attracted to each other. So this is thought to be due to competition for resources. You'll notice they both eat biofilm. Bacteria are usually plentiful, but algae isn't necessarily. So the citronellus get those. The insects seem to be primarily caught by the zaliosus. Again, insects are more likely to be near the surface. Other fish form parts of both of their diets, but the zooplankton are only a part of the diet for the zaliosus fish. Again, this kind of separation in diet and what they're eating is thought to be kind of what drove them to become genetically different from each other over time. Or at least physically somewhat different, and maybe behaviorally different enough. So that brings us to an interesting question. If we're defining a species based on its ability to interbreed, when does that stop? Like, how do you keep things from mating with each other? and producing offspring, because that's the other part of that. So Dobzhansky actually came up with a set of criteria for this, or a set of ideas on how this could play out. This is actually a pretty good study tool. He divided this into two main categories. You have your prezygotic isolating mechanisms, which is anything that keeps the egg from encountering the sperm and fertilization from happening. So some examples of this: the potential mates may live in the same place but not encounter each other, like the fish that live in slightly different depths that we just looked at. They may exhibit habitat isolation. Maybe some of the butterflies live in the tops of the trees and the others live down in the brush under in the forest. I don't know. There may be temporal, time-based isolation. So for example, locusts that we have periodically. There are species of locusts that come out every 13 years, species of locusts that come out every 17 years. They don't encounter each other that often because their cycles don't match up very frequently. So they become genetically separated and distinct from each other. That's a prezygotic isolating mechanism. It keeps them from mating. It keeps the egg and the sperm from getting together. The potential mates may actually interact with each other, but they don't mate. This could be due to behavioral isolation. So maybe they don't find each other's mating songs or mating dances to be sexy. It's just not attractive. That's thought to be a big part of that potential possibility. The individuals may actually copulate. They may go through the behaviors, but the male gametes, the sperm or the eggs, may not be transferred. Humans using condoms are a perfect example of this. The male gametes are released, they just never reach the egg because there's a physical barrier there, and that's the whole purpose of that. And in the last possibility, the male gametes can even be transferred, but the egg may not be fertilized because of some sort of genetic incompatibility that actually prevents fertilization itself from occurring. Okay. So that goes through all of basically the possibilities of when and how you can keep an egg and a sperm from getting together. What happens if they do? The post-zygotic isolation mechanisms occur after fertilization, and these options are a little bit more gruesome. So the zygote may die early during development. The hybrids may be born but not survive. The hybrids might actually even survive, but if they're not sterile, or if they're not fertile rather, they're still considered separate species. So if they're sterile, that's an isolation right there. And the backcross, trying to breed the resulting offspring back to either parental species or to each other, might be inviable or sterile. Basically, an inability to produce viable offspring or fertile offspring at this point. So I have a couple kind of cool examples for you. What we're looking at are two particular species of flower, the Mimulus cardinalis and the Mimulus lewisii. The hybrids of these flowers are not observed in nature. The question really becomes, why not? So they're different colors, they physically look different, but they're actually from the same genus. They're both Mimulus, monkey flowers. Cardinalis, okay, that's like named for the bright red cardinal bird-like color. Lewisii, probably named for the guy who discovered it. All right, what does that have to do with anything? As it turns out, bees actually can't see red. They're blind in that end of the spectrum. Red flowers and colorations aren't particularly attractive because it just doesn't fall in the realm of what they can see. Hummingbirds, on the other hand, are very attracted to red, which is why most hummingbird feeders are made out of red plastic these days, or glass. They actually do sell red-tinted hummingbird food, which is basically a sugar water solution with food coloring. Don't buy that one. Be nice to the poor little hummingbirds. Don't give them the artificial coloring. They don't need it. Okay, so this automatically sets up a distinction between these two flowers and what can pollinate them. On the left, we've got something that looks kind of like it's going to be targeted toward hummingbirds, and on the right, we've got something that hummingbirds don't care as much about, so maybe that's more bee-like. What else can we see in these flowers? Bees actually need a landing platform. They actually have to have this sort of flat-ish, wide enough surface for them to be able to land on the flower and preferentially crawl in and be able to get nectar. So the physical structure of these flowers, if you look at the cardinalis, all of the petals kind of slope down and away from the center of the flower. That's not very bee friendly. Okay, the Lewisii flower, on the other hand, although they slope down and away, they've also got this area kind of around the mouth of the flower that is flat enough that a bee can easily land on that. The other thing that's kind of important on here are those two yellow stripes that you see going down the middle of the flower. These actually act just like runway guides that tell the bee, hey, come on in this way. And surprisingly, they are often UV reflective. So they actually tend to put out UV light signals that aren't visible to us, but that bees can actually see. And Dr. Horth in the biology department here at ODU actually studies those UV cues in flowers. One of the cool things that she's actually found out is that the UV signals in the flowers you buy at like Home Depot are different than the wildflowers that actually grow in nature. So, okay, the Lewisii flower is more bee friendly. The color is even more bee friendly. Bees have better vision in the purple, blue, and violet end of the spectrum, and even ultraviolet end of the spectrum, and not so much red. So right now, this flower is looking like it's nicely set up for bees, and you've got the red one, the cardinalis, that, okay, well, hummingbirds are going to want to come over to it. Hummingbirds don't have to land on the flower. In fact, they can just sort of buzz around, and hummingbirds have pretty long tongues. They're typically maybe like a ½ an inch to an inch in length. They project pretty far out from the end of the beak. Google it. Take a look. It's pretty impressive. So they can actually hover up to the flower and stick their tongue out. They don't even have to like climb or land on the flower. They can get the nectar out of the flower just by hovering near the flower. So that's kind of tricky. Which means that the cardinalis is okay for hummingbirds. While we're on the topic of tongues, bees have surprisingly long tongues relative to their body size too, but not as much as hummingbirds. But this sets up another problem. When the bee crawls into the Lewisii flowers, for example, all of this pollen that you see in here is going to get deposited and coated all over the bee. Great. This is exactly what the flower needs for reproduction. But how is the cardinalis flower going to get its pollen onto the hummingbird if the hummingbird never touches it? That's why you actually see this modified stamen with the anther on the end there. So what happens is as the hummingbird is sort of buzzing around and hovering in front of the flower, this long thing with the pollen on the end of it basically sort of bonks the hummingbird randomly on the head or the body and deposits pollen. It's a way to get around the problem of the pollinator never physically having to touch the flower except with the tip of its tongue. Pretty ingenious. So we have different pollinators. How often do you think a hummingbird is going to go to a Lewisii flower? It doesn't care. Probably not. Similarly, bees aren't going to bother as much with the cardinalis flowers. There's nothing in it for them. They can't get to the nectar. They can't, they're not particularly attracted to the color, and there's not really anywhere for them to land. So, meh. So they have reproductive isolation built in because they use different pollinators. And in the next slide, we get to see even more reasons as to kind of how and why this plays out. Okay, here we have a different set of flowers and a different set or potential set of pollinators. So let's start by looking at flower A, the one kind of up in the top corner there. It is purple. It is fairly broad, and this kind of should be making you think that this is likely to be bee pollinated because that color range should be more attractive to bees than it is to anything else. They like the violet end of the spectrum. As it turns out, the nectar tubes on these flowers, these little projections that you see coming out of the back of the flower, at the base of these is actually where the sweet nectar treat is, and that's what tempts your pollinators to come in. So this is flat enough for a bee to land on. It's got a nice little sweet tasty reward, and the nectar tubes on this are actually relatively short. So the next flower that you actually see is flower B. at the bottom, and it's red. You're probably already thinking hummingbird at this point. And you're probably right, but let's see what else we've got. So the tubes for the nectar on the hummingbird flower are these structures. They are a little bit longer than they were for flower A. These aren't quite drawn to scale. You've also got the modified stigma and anthers that you've got to help deposit the pollen onto your little hovering hummingbird as it kind of flies up and sticks its beak up into the flower to try to get nectar. Yeah, I can't draw a hummingbird's beak. Okay, so far so good. Hummingbirds have pretty long tongues. I mean, on average, I said in the last slide that they're about a half an inch or so, so that's about, yeah, they're pretty decent length. But that's not going to be long enough to deal with flower C over there. Look at the length of those nectar tubes. Those things are gigantic. What on earth could make use of those? That's an interesting question. Moths. So I have a brief story to tell you, and when I was in high school, I was sitting in my kitchen one morning, and fluttering around outside in the bushes was this butterfly, and I watched it for a couple minutes, and it wasn't moving right, and something was obviously wrong, so I went out to check on it and see if it was hurt or whatever, because I'm a sap. And as it turns out, its tongue was totally tangled up in its legs. I know, that's the most ridiculous thing I could ever imagine happening. And you've got to kind of imagine just for a second, how in the world can you get your tongue tangled up in your legs? Well, as it turns out, butterfly tongues are really, really long. So I took the little thing, I scooped it up out of the bush, took it back into the kitchen, grabbed a mechanical pencil, and, pushed out some pencil lead and carefully spent like 20 minutes untangling the thing's tongue from its legs. I totally interfered with natural selection on that. When I got done, it curled back up into this little spiral shape, and it kind of tucked itself not quite in the mouth because it was still too big to fit, but close to. So that way it wasn't all trailing around behind the butterfly when it tries to fly or anything. As it turns out, its tongue was like that long. So we are talking about something that is about 2.5 to 3 inches in length. And as it turns out, that's actually exactly the perfect length to reach down to the bottom of those nectar tubes on this flower. A lot like bees, butterflies and moths tend to land on the flowers. So this one's set up so that all of the pollen up here at the top can actually be deposited when the butterfly lands or when the moth lands on it. And our third cue that this is probably a moth-specific one is actually from the color. It's white. What's that got to do with anything? Flowers that bloom at night are mostly white in color. And there's pretty good reason for that. White can reflect the moonlight because that's really kind of your only source of light in nature. And this is what makes them visible to pollinators. Other flowers like reds and purples, those disappear fairly early because those wavelengths go away once the sun sets. So those flowers become all but invisible, but the white ones almost glow in the dark. They're actually really visible. And so what we have is a night blooming flower with really long tubes. Looks like it's ideally set up for moths. So they actually did some interesting studies, and they actually measured the lengths of those pollen tubes, and they found that bumblebees landed on flowers not pollen tubes, nectar tubes. Sorry, I keep doing that. The bumblebees landed on flowers whose nectar tubes were less than 15 millimeters in length. So that's about that long. Bees don't have super long tongues. Most of the ones that I've seen are maybe about 5 millimeters or so in length. Not that I spend a lot of time looking at bee tongues, but I can Google it and so can you. The hummingbirds took flowers whose nectar tubes were somewhere around 16 to 24 millimeters. So that puts them in the about an inch in length. We're talking about that kind of size. And the hawk moths took the flowers whose lengths were 30 millimeters up to about 70 millimeters, almost 3 inches long. So there was a clear partitioning between the different pollinators for these flowers based on their size. Yeah, there's a little bit of overlap, especially for the bumblebees and the hummingbirds, but Not that much. So, my next question for you, is this a pre-zygotic isolation mechanism or a post-zygotic isolation mechanism? Okay, next up we actually get to look at snails. And the Euhadra snails, we've actually seen these guys before. Some of their shells coiled to the left and some of their shells coiled to the right. Right was actually the dominant characteristic, so homozygous dominant or heterozygous, it's a simple Mendelian trait. What's interesting about these snails is that they can only mate with other snails whose shells coil in the same direction. And this is actually kind of weird. I saw on the internet just a couple weeks ago, there was this thing about find this poor lonely snail, Jeremy, a mate, because his shell coiled left and nobody else's did, so he couldn't have little snail sex. Snails are hermaphrodites, or at least these are, so they actually possess the sex organs of both males and females, but they actually have to mate face to face and sort of backwards. And so if their shells coil in the opposite direction, they can't make the body parts line up right, so it physically doesn't work. This has led to reproductive isolation and in a lot of cases, speciation. So in this phylogenetic tree, what we've actually got are a whole bunch of sister groups that are all either left-handed or right-handed. Blue is right, so this is a group of right-handed snails. This group right below it is a group of left-handed snails. You've got a bunch of righties, a couple other righties as individual species. But since they can't mate with the opposite direction snail, they're kind of limited. My next question for you in this chapter, why were these snails important before? We saw them back in chapter 7. Good luck. Okay. So we've looked at a bunch of pre-zygotic isolation mechanisms that had to do with speciation due to geography, that had to do with, whether the snails could mate face to face or not, pollinator differences. We talked about locusts. So all of these things kept the egg from meeting the sperm. There are other additional considerations though, and this gets into issues of genetics in some cases. So changes in the numbers of chromosome sets that you have, ploidy numbers, this occurs if you've got different numbers of chromosomes. So we're talking about whether an organism is diploid or triploid or whatever. The arrangement of those chromosomes may also matter. For example, humans have 23 pairs of chromosomes, chimpanzees have 24 pairs of chromosomes, and part of the reason that we're genetically incompatible is due to that difference. During mitosis and meiosis, the chromosomes just can't align properly, and the cell has fail-safe mechanisms that should detect that and go, wait, I don't understand what this thing is, I'm obviously messed up, I should not continue to try to make an organism because it wouldn't be a thing. This isn't always the case. Donkeys have 62 pairs of chromosomes, mules have 64, or horses have 64, sorry, and the mules actually have 63. They're actually infertile. They can't breed with horses or with donkeys or with other mules. So this is actually why we consider donkeys and horses to be separate species. This isn't a perfect system. You can get things that interbreed inappropriately, and in plants it's especially common. Plants, in fact, typically survive it well, but animals don't. However, what we often observe, especially for animals, are dysfunctional gametes, sperm that can't swim, eggs that aren't viable, reduced rates of recombination and meiosis, so very low genetic fitness, very low ability to survive or reproduce in that environment. So there's additional considerations. If you look at lions and tigers, which we broadly consider to be separate species, they each have 38 pairs of chromosomes, and they can interbreed. They can produce ligers and tigons, but those are usually infertile. So the question becomes, why does this happen? And there's two major explanations for that. Dobzhansky-Mueller incompatibility, which has to do with genetics and how your genes can interact with each other, epistasis basically, and Haldane's rule, which pays particular attention to the sex chromosomes X and Y. So, as I mentioned, in plants, polyploidy is common, but it also does lead to speciation. So what you're actually looking at basically is the frequency of speciation for a bunch of different types of plant lineages. In particular, if you look at the asterids, about 12.5% of them are polyploid, greater than diploid. For the rosids, it's 26%, or 20%, sorry. For the monocots, the baseline monocots, it's almost 26%. For the higher monocots, it's about 21%. If you compare this to what you see in like ferns and lycophytes, the incidence down here is also pretty high, ranging about 20 to 34%. This does lead to speciation events within these groups. The gymnosperms, ironically, have approximately 0 polyploidy. Their life cycle is actually pretty different. It's almost never happened. It's not survivable for the gymnosperms. So the additional genetic reproductive isolation mechanisms, we're going to start with the Dobzhansky-Mueller model, and this one is actually about genetic incompatibility on kind of a deep cellular level, let's put it that way. So we start off with one organism, and we've just got kind of a prototype cell. It's got 2 different genes, A and B, and let's say that in one population, you get a mutation that creates the A2 allele. Nifty! A2 is an awesome pro-survival allele. It gets fixed in your population because it's such an awesome allele. Yay! You get a gold star. Cool mutation. In this other population, you get another mutation. B mutates in this other lineage. And B2 is an awesome mutation, and B2 gets swept to fixation. And this population also gets a gold star for genetics because, yay, cool mutation that was great for survival. So you would think that when you put these 2 lineages together, that the combination of A2 plus B2 should give you a shower of gold stars. No. What it actually gives you is dead. Oops. These 2 genes, although individually they're awesome, when they're together can be lethal. And so that's actually what Dobzhansky and Mueller were trying to get at in this particular situation, was that sometimes individual genes, when they're combined, can actually have negative consequences for selection. So we actually have a good example of this that comes to us from yeast. They tested this, and the ancestral strain of yeast had 2 particular genes, and those were PMA1 and MKT1. And these were partially responsible for ability to survive in different salt concentrations. So they took one group and they grew them in high salt concentrations, the group with the PMA1 gene, and over time they got a mutant version of PMA1 that they called PMA1 prime. Okay, the prime designates it's new, it's a mutant. Those did really well in the high salt concentrations. The other strain of yeast they raised under low glucose conditions, totally different thing. And these had the MKT gene. And they also got a mutation which they called, guess what? MKT prime. Great, or MKT1 prime. So when they tried to make these 2 strains of yeast together and get offspring from them, the ones that had a single copy of a prime gene, one of the mutants, those survived great. Yay. Good fitness. The ones with PMA1 regular and MKT1 prime, those were nearly perfect in terms of fitness. So that was okay. The PMA1 prime and the MKT1 regular, those did fine as well. So either no copies of the new mutant genes or 1 copy of either mutant gene was good. But when you put those 2 genes together in combination, the yeast took a major fitness hit and they were only about 60% of normal. So not as survivable. Definitely something that natural selection is going to try to eliminate. Haldane's rule. Haldane reasoned that, especially for sexually reproducing organisms, the X and Y chromosomes that determine gender should play a very important role. And he's not totally wrong about this. So he actually noticed that when you're looking at hybrids between two different species, it's usually the male that's infertile, the one that's XY, and the female actually in a lot of cases was okay. And that's kind of an odd observation. So he did some more investigating, and it turns out that we can't keep it as simple as male and female because chromosomes and gender aren't quite as clear as we'd like them to be. For birds actually have X and Z chromosomes, and the heterogametic individuals, the ones with different mismatched chromosomes, those are the ones that are female. The birds that are basically, I think it's ZZ, those are the ones that develop as male. I'd have to go back and check, but close enough for now. And in butterflies, you have a very similar situation where basically XY or XZ or whatever designation you use for butterflies, that also tends to result in female. So for butterflies and birds, things are kind of backwards from what we normally think. So this is why we have to specify the heterogametic individual, the one with the mismatched set of chromosomes, the XY critter, is the one that should be the one that has the reproductive disadvantage and is more likely sterile. Okay. So we've looked at this. Since Haldane came up with this idea, it's been investigated in birds and butterflies and mammals and fruit flies, and it's true something like 97.6% of the time, which means Haldane was really onto something. So looking at this specifically in Drosophila, what we are looking at is where males are heterogametic. You've got 114 hybrids. Of those, 98% fell into Haldane's rule. It was the heterogametic male that was sterile. And if you look at inviability, again, they had 17 hybrids in this case. 76% of them were non-viable. And again, that met really well with Haldane's rule. Not quite as well as sterility, but close. In mammals, it was 100% of the time for sterility and 100% of the time for inviability. Wow, that's really clear cut. When you switch over and you look at butterflies and birds, the lepidopterans and birds, they had fewer hybrids. So when they looked for sterility, 94% of the time in butterflies, 89% of the time in birds. The heterogametic individual, in this case the female, had the reproductive disadvantage. In terms of viability, Haldane's rule was confirmed in 85 to 99% of cases of those as well. So Haldane had definitely discovered something kind of cool. Our sex chromosomes carry really mission critical genes, and if you try to crossbreed between different species, something doesn't work well in that system. This is kind of where we get to look at our last example, the ligers and the tigons, crossbreeds between lions and tigers. So the liger in particular is a cross between a female tiger and a male lion. The order of the parents actually does make a difference. Ligers are actually bigger than either parental species. They grow to be larger than tigers or lions, so that's a terrifying size. The people in that picture are normal height, and that's them walking with a liger. The females are usually able to reproduce. The females are actually XX, so Haldane's rule works here nicely. The males are pretty much always sterile, so that's why they can't produce viable offspring that are also fertile. The reason they grow so big is actually because they are supposed to inherit some growth factors or growth factor inhibitors, and these are missing in the female tiger. So they grow and they grow and they grow and they grow and they grow to these huge sizes. You'll notice that their coloration is really light. It's mottled. It's not, they don't have classic tiger stripes or anything like that. They also have health issues. And yet, ligers are actually the more common crossbreed. Tigons are even more rare. So a tigon grows to normal size. It's a cross between a female lion, which does have those growth factor inhibitors. So basically this is plus inhibitors and this is minus inhibitors. And the male tiger. So they grow to normal lion size. The females can still reproduce. So yay! Again, Haldane's rule is good. So the fact that we see Haldane's rule in both of these particular crossbreeds is kind of cool. But we also see evidence for the Dobzhansky-Muller incompatibility in this system of the growth factor inhibitors that are missing. These genes lead to severely compromised animals in the case of the ligers. There's, I think, if I remember correctly, vision issues, there's health issues, cardiac issues. Their pigment and their coats are mottled, and they tend to be relatively unhealthy animals. Both crossbreeds actually have occurred. They tend to be more common in captivity, in matings that occur in captivity. But ligers have been observed a couple times in the wild. Not very often, but it does apparently happen. Tigons, not so much. They're much harder. You cannot take a liger and a tigon and mate them together. You will get nothing. Those do not survive. Ligers and ligers generally can't produce offspring. Well, that's because the males were sterile. Tigons generally cannot produce offspring, again, because the males are generally sterile. But even if you try to breed a liger back to a female tiger, limited success. And the same with the tigons. If you try to breed them back to either a tiger or a lion, limited success. So there's something genetically, fundamentally incompatible in these species. The fact that it works at all is just kind of a surprise. It's a neat example. So, is the rate of evolutionary change this long, slow, steady process, or does it go in kind of spurts, fits and spells? The first model of slow and steady change, that's called phyletic gradualism, and it came out of Lyell's ideas, and this is kind of what Darwin was thinking about when he was thinking about evolution. Within this, we've got kind of two ways that speciation tends to sort of work, beyond the allopatric and the prezygotic and all the stuff we've talked about so far. Cladogenesis is a branching pattern of speciation, so it's the one that we've kind of been thinking about all through the whole semester with the phylogenetic trees, where one species branches off into two subspecies. But it doesn't always really work out that way. There are other systems that can actually happen. Anagenesis is the other condition where you start off with basically one species, and over time it accumulates genetic changes and becomes indistinguishable for a little while, and then it becomes a different species, and that may sort of kind of morph into another species, which morphs into another species, until you end up with something that's genetically and physically very distinct. But it's just through gradual changes without any true extinction event or true branching event that takes place. It's just gradual morphing. So both of these can actually happen. And we saw examples of this in the horse lineage, both cladogenesis and anagenesis. We saw the branching part, particularly in the Miocene, where we had that adaptive, bunch of lots of branches that popped up. But we had also seen areas below that part where one species just kind of gradually changed over time and became something different. The next question is, how long does this take? Is this like a over eternity kind of a process, or does it actually change the rate from time to time? And so this is a newer idea. It's called punctuated equilibrium. And what this is periods that are mostly slow and steady change, like we've all been traditionally taught, but with short periods of very sudden, rapid change interspersed in there. So, going along fine, boom, lots of new species form. Going along fine, going along fine, going along fine, boom, lots of new species form. I'm so good at art. Okay. We actually see a lot of evidence for this, and in particular, this seems to occur when you have a new environment, a new ecological niche open up, or niche, however you choose to pronounce it. When organisms move into this new environment, they have usually few predators, a lot of opportunities, abundant food supplies, and they rapidly diverge from each other as they kind of fight out for those resources. Sympatric speciation is a really good example here. So in particular, we've seen really good evidence for this after the Cambrian extinction around 500-ish million years ago. Most of the animal groups that ever lived went extinct, and after that there was this huge explosion of new life forms. We see another one that happens right after the KT extinction with the explosion of mammals, as we sort of kind of came up from, okay, I'm assuming you guys have all watched some sort of dinosaur show, right? And they show the little mammals underground in their little burrows. Well, okay, the dinosaurs are extinct. The little mammals come up from underground, they look around, they go, ooh, there's nothing to step on me. They go out and they do what mammals do best, and that's breed. They create lots and lots of offspring, and they populate the earth, and voila, here we are. Okay, maybe not that simple. That is a gross oversimplification, but it's more fun that way. So how do we know that extinction even happens? This one's actually easy to document, really, really easy to document. A lot of examples of extinct animals have been found. Dinosaurs are not the only examples by a long shot. So the short-faced kangaroo, the mastodon that you see there, and the saber-toothed cat are all examples of animal species that used to exist that have disappeared in the last 50,000 years. They're not here anymore, but we find fossils of them. We find examples of them sometimes intact. Other things like mammals, we can get whole mammals out of ice. We've actually found juvenile, like really young mammoths completely encased in ice with fur and everything. I mean, it was really impressive. We find whole animals that have been trapped in peat bogs that are extremely well preserved. But even more than that, we've got modern examples of animals that have gone extinct. Carrier pigeons were used to carry messages in war situations, and they don't exist today. They just died out. We've got a bunch of organisms that have actually died out and disappeared through written human history. Some of them we've found living examples of, which means they weren't truly extinct, but basically the definition of extinction is when all of the members of a species have died out and left no descendants. That's probably fairly self-evident. So where did they go? Well, if you're like me, you were probably taught in elementary school, obviously we hunted everything to death. Great. How did we hunt T-Rex? I'm just curious about this because if I see T-Rex coming, I'm going to run fast. And then I'm going to try to find the scientists that recreated that thing and make them watch Jurassic Park on an endless loop for the rest of their lives. Okay. So other movies that are important in this context are Ice Age. I hope you guys have all watched Ice Age. If not, homework, go watch it. You have to watch Scrat run around and chase the acorn. It's hilarious. But it brings up another point. There are a lot of ways that things can die out. Major climate changes, ice ages, freezing to death is an equally valid way of dying. Predator-prey, overhunting, overfishing, which doesn't necessarily imply humans. That can cause extinction. Diseases can cause extinction. Natural disasters can cause extinction. So all of these are potential factors. Not all of these events where we see extinction things are actually thought to even be human-induced. So, for example, in this particular map and graph and all that stuff, what you're looking at are several different organisms where, okay, you've got the red bar, which is when humans arrive in an area, the little blue window is climate change, and the perceived cause of extinction, what we can attribute that to, humans are red. So, okay, we probably killed off the ground sloths. But it's very unlikely that we actually killed off the mastodons. Oh, well, no, actually, wait, I take that back. Very likely that we did. Okay. But those critters, not so much. they were pretty much already going extinct by the time we came on the scene. So that was not our fault. Some of these others, jury's still out. We just don't have enough data. So humans have probably contributed to some of these extinctions, but probably not all of them. And it may be a combination of multiple factors. Climate change, global warming, global cooling, human activities, diseases, predators and prey, all kinds of things that can happen. Volcanic eruptions, hurricanes, whatever. There are a lot of potential interacting factors. There's a big argument going on the internet and in the scientific community right now about whether or not humans are causing a major sixth mass extinction. And that's actually part of your homework for this chapter. I want you guys to go read online and think about what you find out about the thoughts about the sixth mass extinction versus what you're learning about what a mass extinction is in this chapter. So we'll come back to that. So with regard to extinction, we've got two main categories. Background extinction. This is the normal day-to-day extinction rate, and this is a fairly typical process. In fact, if you think back to earlier this semester, when we looked at Darwin's original notebooks, he actually had written in there that he thought extinction was required. It was actually necessary to keep the tree of life functioning and healthy. So the background extinction is composed of endemic species going extinct. These are small, locally dwelling species that only existed in one region, and other small individual types of extinction events. They affect one or maybe a few scattered species here and there. This can be due to predators, it can be due to human activity, habitat destruction, all of those things that I've already mentioned several times now. The mass extinctions are a totally different thing. These tend to be sort of periodic, or at least that's the thought, and they affect somewhere around half or more of all species across multiple major groups. So not just all the chickens. We're talking not even all the chickens and all the vultures. We're talking the chickens and the trees and the frogs and the bees and the plankton in the seas and the, I don't know, the grass outside your door. So lots and lots of different things being affected. It has to be much more broadly based. These are generally attributed to major climate changes or catastrophic events like meteor impacts. Some of them, we just don't know what caused them. We're going to talk about two in particular. And then, like I said, your homework, go investigate whether humans are actually causing number 6. So what leads to extinction and how this all works out, it's largely dependent on food webs, this concept that you learned back in like elementary school sometime. So in this particular example, humans brought pigs to the Santa Cruz Island because, well, I guess we like ham and bacon. I'm just being politically incorrect there. But why else would you bring pigs? They are a good food source. Some of the pigs escaped and formed wild populations. Those attracted another native species, the golden eagles. These were endemic. They were native to the Santa Cruz Islands, but they weren't really very common. However, they got lots of pigs to eat now, so their population kind of went through a boom cycle. It expanded dramatically because they had all these pigs to eat. Yay! Okay, when it got to the point where there were too many birds to eat the pigs, they actually also ate the native foxes, which was their natural prey. These foxes mostly feed on mice and insects and fruit, which is kind of strange. I don't usually think of foxes as eating fruit or even insects. Mice are okay, but bugs? Fruit? Actually, thinking about that, if you guys have ever tried feeding your dog fruit, you'll find that there are some that they actually like. Some of my students in my face-to-face class have mentioned strawberries, apples, tomatoes, watermelon, cantaloupe, pumpkins. Pumpkins tend to be very popular. A couple of years ago, I went to Busch Gardens for Howl-O-Scream, and they actually had pumpkins that they put out for the wolves that they had on the premises. It was really cool. I got to watch them play with the pumpkins and eat the pumpkins. Even big cats like pumpkins. There's YouTube videos. Go check it out. Okay. So the eagles ate the pigs. When they ran out of pigs, they continued eating the foxes. Since their population had gotten really big, the now oversized eagle group ate all the foxes. Unfortunately, the foxes also kept control over the skunk population. And this is the western spotted skunk that is also native to the Santa Cruz Island. And since its predator, the fox, is now gone, there were lots and lots of skunks. And they're a pest problem. So because humans brought pigs, they ended up with a bunch of extra skunks. Oops. Unforeseen consequences. Food webs. This does actually tie together. It's all about predator-prey interactions. You don't necessarily have to have an extinction event, but you do see a lot of changes in these population levels. As these endemic species rise and fall in numbers, particularly as they fall in numbers, if those golden eagles all die off, they're lost from the world because that's the only place that they're found. So extinction among endemic species is particularly problematic, and one that we tend to worry about a little bit more. This is mostly critical when a predator relies on a sole primary food source and doesn't really have a lot of other foods that it tends to eat. Okay, this example actually, for me, the story begins back in 1996 in my freshman year of college when I was taking biology for non-majors. One of our textbooks, I should put that in quotes, was called Tracking the Vanishing Frogs. And what this was about was a crisis that was affecting frog populations across the world, and we didn't know what was going on. And that top picture that you see with all the little dead froggies in it, this was being observed everywhere throughout the world. And it was really, really scary because we didn't know what was triggering this. We didn't know what was causing this. Frogs are actually used as an indicator species to kind of look at the health of an ecosystem because they do so much transport back and forth across their skin. They absorb things from the environment and they use it to get rid of toxins and expel waste products through their skin as well. So seeing all these frogs dying off, our immediate thoughts as scientists and what we were discussing at this time was, well, obviously there's been some sort of, I don't know, heavy metal leakage or something, some environmental poison or toxin has gotten out and it's killing all the frogs, and oh my God, are all the frogs going to go extinct? And there was actually a lot of discussion about this. It was kind of an interesting topic. So I was really excited to actually see that revisited in this textbook. It made me feel really kind of happy, and I finally got an answer to the question. So in those maps that you're looking at, what you see in the green map is actually the amphibian diversity in the world. So you'll notice that South America has lots and lots and lots of amphibians, a lot of frog species. The US, we're really small and skinny and kind of compressed, so nowhere near as much. Africa also has a fair frog population. It's represented as fairly large, although the diversity index, kind of the numbers of different types of amphibians, aren't as high as they are in, say, South America. That's still better than what you tend to find in, Russia. Asia has a fairly good frog population based on the numbers that you see there too. Compare that map to the one that you see below it, and this is where you find the numbers of critically endangered and threatened amphibian populations around the world. You'll notice that it's actually North America that's hit pretty hard. Africa has some. It's kind of almost in like hot spots. And this Asia also is experiencing kind of a lot of amphibian loss. So is Australia, which is kind of interesting. And so these areas don't match up perfectly. But what they were observing was that among the surviving frogs, they tended to be lethargic and anemic. They showed damage to the liver and the skin and other organs like the kidneys. So all of this kind of was pointing toward maybe some sort of environmental poison or something that was killing these frogs. Part of the initial piece of evidence was that this would also pass to healthy frogs. So that kind of made us start thinking that it might be an infectious agent. Moreover, as this was transmitting to healthy frog populations, in Australia in particular, they were actually able to track it as it moved up the coast of Australia. It actually migrated northward at a rate of about 100 kilometers per year. So in 1978, there had been no outbreak, but this really got started in the mid-1980s or so, and it moved northward basically over the next several years. And when I first learned about this was a couple years after this graph ended. That was 1996. But there was still a lot of speculation about what was going on. Now, if you're like me, you've watched at least a few episodes of House or some other medical show, so whenever you think infection, you immediately go, bacteria, ah, or viruses, ew. Okay, that's what we thought too. So we started trying to identify the bacteria or the viruses or the environmental toxin or whatever was poisoning and killing all these frogs, and we couldn't do it. Nothing fit. And it was still happening. And eventually it was actually linked to a fungus. And in particular, this is the Batrachochytrium dendrobatidis fungus. Don't ask me to pronounce that one again. It's abbreviated BD for a reason. The areas where we find this fungus in particular are the US. We see it in the continental US as well as in Alaska. Australia has a lot of it. There are some samples that are found in South America, but it's not as prevalent, but very little in Africa. And this matches up very well with the extinction rates that we were looking at in the amphibian endangerment from a couple slides ago. And it was finally this fungus, chytridiomycosis, that was linked to the declining populations of frogs. It actually affected their ability to transport chemicals across their epidermis. So it was tied to their skin, just not in the way we expected. And to this day, since I've learned about this and a few other things, you've got to keep in mind that fungi can also cause disease. We don't think about it as often, but when I watch some of those medical shows, I find myself screaming at the TV sometimes going, "Did you check for fungal infections? You've tested for all the bacteria and all the viruses, and now you're going to go back and redo it? Well, what about, parasites? What about fungi? What about other things that can cause illness?" Yeah, that it gets overlooked a lot. So this was a really cool example, and I was really excited to finally actually get an answer to what was killing off the frogs. Incidentally, it was something that I had even observed. We had a small pond when I was growing up, and over the last few years before I started college, we'd had fewer and fewer spring peeper frogs. We used to be able to go out to the pond every year in the spring, and we'd find these masses, like giant arm-sized masses of frog eggs, and you could actually watch the little tadpoles develop and then hatch into little baby frogs, and I loved doing that. But over the last few years before I started college, there hadn't been as many, and a couple years there hadn't really been any observable frog eggs in our pond, and I had always been disappointed by that. So now I know it was a fungal infection. So that brings us to the mass extinctions. What makes the frogs not a mass extinction? It's only affecting one group, one type of thing. It's just the amphibians. It's not the frogs and the bats and the trees and the cats and the, oysters in the sea. So that limit means that it doesn't really qualify as a mass extinction, even though it's affecting many different frog species. In fact, lots of different frog species. Today we've also got a problem with bats and the white nose fungus that is spreading across the US and wiping out bat populations. Bats eat mosquitoes, just keep that in mind. So neither of those qualify as mass extinctions. They're caused by different fungi. Although they're affecting lots of different species within these groups, it's still not widespread enough, and it's not affecting a big enough diversity to be qualified as a mass extinction. So just to reiterate, what we really need is something like more than about half of multiple major genera, genuses. A huge loss of diversity across different taxonomies, different morphologies, so different shapes, different sizes, different kinds of things. All of this has to be affected at levels greater than 40 or 50% of the species. Typically, there's about 5 major ones that everybody agrees on, maybe 3 others that aren't as well agreed upon. But our 5 major mass extinctions to date are the Ordovician, the first one that we've got evidence for in the fossil record, the Devonian, the Permian, which was particularly severe. It was thought to be the worst of the mass extinctions. We'll talk about that one in particular. The Triassic and the Cretaceous, that's actually the K of the KT boundary part. the one that occurred about 65 million years ago and ended the dinosaurs. And then, are we number 6? So even after a mass extinction event, let's say half of all the life on the planet goes away and dies, 10 to 20% of the residual species, the stuff that initially survives, never really thrives in the subsequent time period. And we call those dead clades walking. They never really recover and they subsequently go extinct. They just they made it through, but they squeaked by just barely. You know, they got a D- in the survival thing and they just didn't make it, even though they survived that initial extinction event itself. So we call those dead clades walking. They often give us kind of a misestimate of actual extinction rates and survival during these events, but we can correct for that to some extent. So here's kind of what we're looking at. In terms of the mass extinctions, the ones that I just listed, we had the Ordovician. That one occurred around 435 million years ago. The Devonian at 355 million years ago. The Permian, the really big one, at 250 million years ago, followed shortly by the Triassic at 203 million years ago at that one. And the most recent one is the Cretaceous at the KT boundary. So when a lot of people look at this, what they see is kind of this pattern of extinction, extinction, time, extinction, extinction, time, extinction. Oh wait, where's our second little peak? There should be another dot in here somewhere. And there isn't. So a lot of people think we're actually overdue for another major mass extinction. Now, based on the data that there actually is here, I'm going to mark this one out just so you know it's not real. There's actually 1, 2, 3, 4, oops, 4 is that one, 5. 1, 2, 3, 4, and 5. People think they seem to occur kind of as a doublet, about every 100 million years or so. So this begs the question, are we due for a major mass extinction? Is it right on top of us? Are we overdue for one? Is something going to come wipe us out? Is it our time? Okay, here's what I honestly think about that. I am not convinced that that's actually a pattern. There's only 5 data points. That is not enough for me to be really convinced that it's a trend. I can flip a coin and have it land on heads 5 times in a row. That's not convincing yet. So maybe and maybe not. It's really kind of hard to tell. And I suspect what causes a lot of that doubleting effect are those dead clades walking. Because, okay, so you had the animals that survived the Ordovician. They should have gone extinct during the Silurian. That was a pretty brief period. But we see another one that's kind of on the border not too long after that, Devonian and Mississippian. The end Permian, the very next geologic era, there was another extinction event. So those may have been that 10 to 20%, those dead clades walking. Because we didn't necessarily see that after the KT boundary event, we're not sure. Is something coming to get us? Is there going to be a worldwide plague of zombies to wipe us out? Are we that seriously overdue that nature has to somehow correct it magically? No, not necessarily. A lot of scientists do think that we're overdue for one. The pattern in the fossil record looks like a pattern to a lot of people. But like I said, 5 data points, I'm not totally convinced. So what makes a species more likely to survive an extinction event? And this is an important question. So what we've observed is that the animals that went extinct during these major events, most of them had higher levels of oxygen use. They were bigger animals, they were more complex, they were more aerobic in nature. So the animals and the species that require less oxygen in their environment tend to have better survival rates during any extinction event. And that's also somewhat true during background extinction events too. Animals that hibernate were more likely to survive, which actually makes a lot of sense. If you can go curl up in a hole and sleep through the zombie apocalypse, that sounds like a pretty good way to survive it to me too. Keep in mind, when animals hibernate, their metabolic rates usually drop lower, their oxygen consumption goes down, their body's metabolically slow, so a lot of those processes that require inputs of energy aren't as bad. So they can come up periodically, maybe find some food, and hopefully go back to sleep. That might actually be really useful. Unfortunately, we can't do that. So that increases our chances of going extinct during the next cataclysmic event. Burrowing and living underground improves your likelihood of survival. That one we could learn to do. Part of the reason for this is because once you're under 3-ish feet of soil or so, temperatures stabilize, humidity levels stabilize. The environment is a little bit more consistent when you're underground. It also means that you've got this nice layer of soil blocking out radiation, filtering out, I don't know, volcanic dust or atmospheric poisons or whatever it is that's triggering that extinction event. It's less likely to get to you if you're underground. Another factor that is often considered is longevity, and this really isn't how long an individual lives, like whether I live to be 70 or 90, it's more about how long has the species already been around. And cockroaches, let's use that as an example. They've been around since at least the dinosaurs and probably even earlier than that, and they haven't changed very much. So does that mean that they should go extinct because they've already existed for like their frame of time? The answer to that seems to be no. This is actually one of those issues or one of those factors that is actually not a determinant of whether a species is likely to go extinct. So longevity, how long it has existed, does not adequately predict whether it will go extinct the next time around, except for the dead clade walking issue. Geographic range, how much territory a species takes. Humans are spread pretty much all around the globe. So let's say a comet smashes into North America. Okay, great. We're all dead. That sucks for us. But there's other continents out there. There's South America, there's Africa, there's Europe and Asia. Those people probably are not going to be as heavily impacted, and there's a better chance that at least some of that population, some of that species, our species, would survive in those cases. So the more geographic range a species takes up, generally the more likely it is to survive a major mass extinction event. That's not always true, but it is, at least it does seem to give them a better chance of survival long term. So the first one that I get to talk to you about is the KT boundary. The reason we all talk about the KT boundary is because it's actually just the most recent mass extinction event that we've had. And like any crime scene, the evidence is still freshest from this one. The older they get, the more trampled they become, and the less you can get out of them. So this one is the most recent, even though it's still 65 million years ago. So in this particular section, we're going to look a little bit as to the evidence that we actually have for this and kind of what it tells us about what went on. So the KT boundary actually refers to a particular layer of rock that you can actually see there in the images that's very rich in iridium. And this is an element that is common in asteroids, but isn't really part of, or a large part anyway, of Earth's normal geology. And this layer is actually found at all of those red dots on the map, pretty much all around the world. So everywhere we look, we see this particular layer of rock, and this is kind of mind-boggling. What could have happened that would have been so big that it could have caused this one layer of rock to change all around the world all at once? That's bizarre. And why is it so rich in iridium? Well, this is one of the first things that makes us think that it was probably an asteroid impact, because we do see this in space rocks. But there's actually a bunch of other pieces of evidence that support this idea. So that's what we get to deal with next. If it was a meteor or an asteroid that smashed into the planet, then we would expect to find an impact crater. So this is what impact craters look like. You've got the Pretoria Salt Pan Impact Crater and Arizona's Barringer Meteor Crater. Now, the Arizona crater, I don't know if you guys can see this, but way down in the bottom, there's that little white streak that kind of looks like a path, and then there's this little white dot sort of right in the middle there. That's a building. If you're standing on the edge of the meteor crater, you actually cannot see people walking around in the bottom of that crater. It's that big. I've never been myself, but it's on my bucket list. Okay, so an asteroid big enough to make that crater was 150 feet in diameter. That's a pretty big falling rock. The KT asteroid that had to coat the entire world in iridium dust and leave a layer of rock, that was estimated to have been 218 times bigger. We're talking massive, really, really massive. That's huge. That's like, oh my God, how did the planet not just break in half? I mean, it's still a surprisingly small area for the surface of the Earth. But people then ask the next question, where did it hit? Shouldn't we see a crater? I mean, we've got the Barringer Meteor Crater there, and we've got the Pretoria Salt Pan, and there are lots of other impact craters. So where is this 218 times bigger crater? Okay. Let me ask you another question, and I'm not even going to put this one up as an actual question. This is just a rhetoric question. How much of the Earth's surface is land? Rough percentage. Okay. In my class, they tell me that typically they're taught that it's somewhere around 25 to 30%. Cool. So that means that the other 70 to 75% is water. Therefore, based on probability, it's a lot more likely that the meteor or the asteroid or the whatever would land in the water. So we should be looking in the oceans since we haven't found it on the land. So since somebody got all logical about it, they've actually been doing that. And right now, the best evidence that we have is for a crater called the Chicxulub crater. And it's just off the Yucatan Peninsula. So if you're looking at, and I'm not good at art, so okay, here we've got the coast of the United States, there's the Florida Panhandle, Louisiana, you've got the Texas thing going on over here, this is the Gulf of Mexico. As you come down into South America, you've got this little finger of land that sticks up, that's the Yucatan Peninsula. The Chicxulub crater is actually thought to be right around in there. It's big enough. It's just underwater. Or most of it is. Okay, so if we think we found the crater, what other evidence do we actually have that it was probably an impact from an asteroid? Here's what we can make some predictions about. If this is going to have to be a huge, huge asteroid, even if we never find the actual crater, we can predict that it had to be really big. We have about 7 right now that are orbiting the Earth that are 10 kilometers in size or bigger. They tend to smack into us about every 30 million years or so, so we are actually overdue for that. Okay, aside from the huge crater issue, when this thing hits the ground, whether it's ocean or whether it's land, it doesn't make much difference. It should kick up a huge cloud of rocks and dust and debris and ash. This is going to be a flaming ball of destruction coming into the atmosphere and through our sky. So what we actually would expect to see when something like that happens is that cloud of ash and debris that's generated from this thing should actually block out the sun. Now you're probably wondering, how in the world would we know that? And what on earth would make us think that photosynthesis would stop? I mean, that's a bit of a stretch. Well, here's what we do know. In 1883, there was a major volcanic eruption, Krakatoa. And we actually saw a cloud of ash and debris from this volcanic eruption that was only 18 cubic kilometers in size. This actually surrounded the Earth. People actually observed the darkening of the skies, and there was a global drop in temperature for about 2 years after this. We were measuring temperatures. We had thermometers by then, and people were recording that. It didn't have a huge impact on extinction, but there was a noticeable drop in photosynthesis, and that's easily documented by a loss of pollen. If the plant can't make energy from sunlight, it doesn't have any to make plant reproductive bits, so less pollen. We can actually track that for the KT event at 65 million years ago by going back and looking for pollen granules in rock and fossils, ice core pollen, and try to see if it's consistent. So far, it has been. So if your photosynthetic stuff dies, again, you know, third grade elementary school food web stuff tells us that when the plants die, the herbivores die, and the carnivores die. So this results in a large loss of lots of different species, affecting a huge amount of the world, and a basically mass extinction. And we saw a fair amount of this just from a volcanic eruption. A large one, admittedly, but still. So what else do we know? Those are things that we can make some predictions about, and then we can go test. We can go look for evidence of food chains collapsing and pollen decreases and ash and debris that has been spread worldwide, like all that iridium. But here's what else we actually also find. We find some really bizarre amino acids, and these aren't like the ones that you see in the codon table that we looked at earlier. These are things like isovaline and alpha amino isobutyric acid. Living things on Earth don't make these. We do find them in asteroids, though. So that's actually another pretty good piece of evidence. We do find these in the KT layer along with all that extra iridium. So more extraterrestrial stuff. There are some glassy mineral formations called spinels, and these form in response to high temperature, heat, and pressure. We also see impact diamonds. These are literal diamonds that form during these high pressure, high velocity, high speed impacts of meteors and stuff crashing into the Earth. They've been observed in response to much, much smaller meteors and asteroids than what we would expect to have seen for this. So wherever we're looking, we need to find these things. We need to find mineral formations like the spinels. We need to find impact diamonds. And secret story for you guys. I told my boyfriend that if he ever got me a diamond engagement ring, I wanted it to be an impact diamond. Google them. They're not actually all that pretty. I just think it would be all sciencey and geeky and cool. And much more unique. So that would be me. We actually do find these in the Yucatan area around the Chicxulub crater. That doesn't necessarily mean smoking gun, hey, we found it. It just means there was a really big impact here. We do see that there was a huge loss of oceanic life that corresponded to this. So it wasn't just the dinosaurs. It also affected critters in the oceans. There were like 3,000 fossils from over 340 distinct genuses of bivalves. So a lot of oysters, clams, those types of critters also disappeared. And they're near the bottom of a lot of food webs. They're filter feeders. So whatever was going on, it created a huge problem worldwide. Last but not least, we've also got this additional piece of evidence that comes from a totally different branch of science. Astrophysicists have been looking at what's been going on with the planets and the asteroid belt in our solar system, and they've found these big giant chunks and fragments that look like the remnants of one that had broken apart, and they calculate that this probably happened just before 65 million years ago. So there's a fair chance that our space rock that smashed into our planet and killed everything, well, not everything, but the dinosaurs, might have actually been generated from a larger piece of chunking rock floating around in our asteroid belt. Kind of a scary thought. We do know those escape, and we do know that they, orbit the sun and the Earth, and they get thrown out into the solar system. So that part alone wouldn't be surprising. This was still actually almost a minor event after the end Permian extinction. This is the one that occurred about 250 million years ago. And it's estimated that 90% of all living things on Earth died during this extinction event. And it went on for 7 or 8 million years. So it was not a short, like, oh my God, it happened in a week and everything died kind of thing. It persisted for quite a long period of time. Its biggest impact was actually on aquatic life, so the oceans were really hard hit, and it was only something like 70% of land life that died out, only. So what you're kind of looking at is during the Permian period, in this particular timeline, these layers of rock actually correspond to the time that we call the Permian. The end Permian boundary is basically where we start seeing Triassic era fossils. But somewhere around one particular layer of rock, it's only about 5 centimeters thick, so that's about 2 inches, almost exactly. This is a small layer of rock, but oxygen levels plummeted, and we see almost 94 to 96% of marine species, ocean-dwelling species, that vanished. They just died off. And so in the subsequent layers of rock, layers 26 and 27, we see a lot less life going on. But these are ones that actually persisted over the next basically 7 to 8 million years. When you compare these to what we actually saw before that event, before the oxygen levels tanked, you've got a lot more structures. You've got a lot more diversity in this layer 24 before the oxygen levels dropped off. So since it was the oceans that were hardest hit, this implies an ocean problem. Plankton, bottom of the food web. I learned that back in like 3rd grade or whenever it was. So this is kind of what you're looking at, is a before and after snapshot of the types of fossils that we've been finding in these rock layers. On the left, you've got radiolarians that disappeared, and on the right, you've actually got images of rock from after the Permian extinction. A lot less diversity, looks like grains of rice scattered in there, nowhere near the levels of complexity. As our new reefs formed, that's what you're looking at. They were very, very different in terms of their biological composition than the Permian era coral reefs had been. So there was a huge change and a lot of destruction and death. So this kind of makes you wonder, what on earth would kill 96% of things? That had to be a gigantic mother blanking asteroid. Holy bleepity bleepity bleep. Okay. The land species were affected too. It wasn't just the oceans. In this one particular region, this was called the Karoo Basin, they found 2 species of fish-eating amphibians. They died out. 72 species of reptiles, some of which were herbivores, others were carnivores. They varied in size. They all died out. That whole entire ecosystem, the entire Karoo Basin, died. It just vanished. It quit. It was gone. Kaput. There is no more. So you're looking at an estimate of diversity on the right in the figure there of life forms before at the end of the Permian era, even after a lot of this had already started, versus the early Triassic period. And you can see that life was staggeringly reduced. 72 out of 74 vertebrate species were lost. That's us. We're vertebrates. These were our ancestors in an evolutionary sense. That means we all descended from what? The other two? Inbreeding. Okay. 36 out of 48 amphibian and reptile families vanished. Not genus or species, but families. Whole groups of genuses and species. Plants and trees and woody bushes all went extinct. So everything was hit really, really, really hard. Kind of makes you worry a little bit more about the health of our oceans today and keeping our plankton all happy. So this begs, like I said, the question of what caused this. And honestly, we don't think it was an impact. We don't think it was a meteor or a comet. We think it was actually an earthly event, a terrestrial thing. And here's what's kind of interesting. The story on this has actually changed since I took evolution. I took it as a senior in college. Ours was a senior level capstone course. So between the years 2001 and 2011, when I started working with this edition of the textbook, everything changed. Our entire way of looking at this shifted a little bit. And it was so weird to me that I actually was like, wait, what? That's not what I learned. So here's what I was taught. I was taught that basically you've got this map of the world here. Still had the one big supercontinent, Pangaea. So there's a lot of the world that's covered in water, and it's pretty stagnant because there's really not much breaking it up. There's not a lot of ocean currents going on at this point. So as the planet started to warm and as the climate started to change, we actually this is what I was told initially, a lot of the marine life actually started to die, and it would settle to the bottom of the oceans. And basically over time, this built up with toxic byproducts from the rotting and decaying, and it would occasionally burp. And you'd get these big outgassing bubbles of all of this toxic stuff, and that it would poison the living stuff, which would then float to the bottom and exacerbate the problem. And this is what triggered the massive dying off in the ocean. Okay, and that was literally what was written in my textbook. I went and pulled it back off the shelf. Mine actually had very little resale value, surprisingly. So I kept it, and I've still got it even. When we got this new textbook, it actually told us about some new evidence that we have just uncovered in the last 10 years that have changed how we think about this. And in particular, one of the things that was different were these Siberian traps. This is basically a volcanic range in Siberia that is now dormant to extinct. But the evidence suggests that there was this massive, like way bigger than Krakatoa, this massive field of volcanoes that all went kablooey basically at the same time. So when these things erupted, it caused a global blackout again, like with Krakatoa. You got the ash and the debris, and a lot of that settled in the ocean. Blocking out the sun is going to affect the plankton, which is a lot of the living things going on, and the land stuff. So those are the photosynthetic things are going to start dying off, which is going to lead to dying off of other things. But it was more complicated than that. So that's where this flow chart over on the left-hand side comes in. So up at the top, you've got the Siberian traps that erupt. This causes the release of a whole bunch of different gases that are well documented to come out in volcanic eruptions, like sulfur dioxide, rotten egg smell, chlorine and fluorine gas, and by themselves, they're pretty toxic. Carbon dioxide levels went way up. That was also ejected in large volume. So what these things caused was this next tier of reactions in the planet. We got a brief period of global cooling, partially due to the sulfur dioxide. This triggered temporary growth of glaciers and falling sea levels. This didn't help the marine life. The global cooling didn't help the marine life. Life tends to like things a little bit warmer. We do better with some heat. The carbon dioxide emissions then also triggered global warming, which occurred right after the brief global cooling. So this rapid fluctuating change in the temperatures, cold and warm again, didn't help either. Life couldn't cope with the rapid change in temperature, and this contributed to more of the extinction events. The warming also helped the oceans to remain stagnant. It reduced mixing, so there wasn't a lot of oxygen getting stirred into the oceans. That didn't help marine life at all. The chlorine and fluorine contributed to acid rain, which also killed off land plants. As it got into the water, it caused shifting pH to some extent. That didn't help. It also triggered fungi to proliferate, and we actually see some evidence for that, which is kind of neat. There was a huge increase in the number of fungus that we actually can see in the fossil record right around that time. There's increased continental weathering, which is a result of the acid rain and the carbon dioxide. All of this kind of fed in together. We see some chemical changes that are also consistent with volcanic eruptions, such as changes in our carbon isotypes and our strontium isotypes, and the ratios of those that we would normally see shifted as well. So I had to look at this and go, wow, okay, things changed. We got new data. You know what? This actually makes more sense than what I was taught. Cool, okay, new model. Now I understand. 10 years from now, it may change again. I'll look at it. If it makes more sense than what I've got now, cool, excellent, I'll adopt that too. And this is just part of the life of a scientist. But it's kind of neat that just in my relatively short career so far, to you guys it seems long, but 10 years is not that big a span, that things could have changed that much about something that happened 250 million years ago. And we're still just kind of figuring this out. So in terms of your homework question, are we facing a sixth mass extinction? Here's some of the data that we've actually collected so far. What you're looking at are basically the percentage of animals that are threatened or already extinct. And the stuff on top, this is the modern day stuff. So mammals about 22%, birds about 14%, reptiles 29%, amphibians 31%, and climbing, thanks fungus. Some of the fish 28%. Gastropods, like snails, like our euhadra snails, 56%, but it's decreasing. They're actually rebounding. And so are things like bivalves. The cycads, like ginkgo trees, those are at 64%. Those are the gymnosperms, the ones that didn't show a lot of polyploidy. So there might be a link there. This is actually the highest level that we see in any of the groups. Conifer trees, 29%, sharks, 17%, and decapods, like lobsters and stuff, 19%. None of that reaches the 40 to 50% threshold, except for snails, and that's decreasing. Bivalves, we're getting close, it's decreasing again, and the gymnosperms. So based on the definition that we've got so far, this doesn't look really very promising. And if you compare that to what happened during any of the major mass extinctions, everything was 75% or higher. So we've still got a pretty good gap there. That doesn't mean that we're headed in the right direction, and it doesn't mean that we are not contributing to problems that may already exist. But you, my students, need to go forth and read about this on the internet, preferably from some reliable sources, but start with Google and see what you find. Check out like Scientific American. They've usually got a pretty good reputation. And do some comparison reading. Hit Twitter and find out, see what people are saying. And then in the discussion board, in the homework, let me know what you think. I really want to know whether you think the evidence is convincing or whether you think that maybe this is just a normal part of a natural process. So that is the end of chapters 14 and 15.